Welcome to the CompuCell3D Workshop, Module 4.23. We have a great guest presentation by Job Berkhout from Utrecht University in the Netherlands. He's going to present his paper on computational dynamic systems model for in silico prediction of neural tube closure defects. I also want to remind everybody that this is being recorded and will be distributed on YouTube this weekend. In addition, Job's paper won first place in the 2025 Open Virtual Tissues Reproducible Agent-Based Multicellular Model Competition. So welcome, Job.
Well, thank you for the introduction, Alan. So welcome, everyone. My name is Job. I'm affiliated with Utrecht University, and as Alan introduced, I will present the publication towards a virtual embryo, a dynamic system model to predict neural tube closure defects. So birth defects happen way too often. With 1 in every 33 live births in the US, there is a defect. And as you can see on the slide, they are also very severe. In this case, if you see severe skull malformation, or the back's completely open, or the lower back, for example. And these can be the cause of different chemical exposure. So when we are living our lives, we are exposed to chemicals, many different chemicals every single day. Say the food that we eat, or the air, the pollution in the air, or maybe various household items within our cabinets. All of these chemicals can be a risk for a potential neural tube closure defect. And there's currently not really suitable testing strategy available. Of course, we have a rat as an animal model, but that comes with different ethical concerns, as well as there's really a need for a greater chemical coverage. Rat studies are very slow, they're expensive. It's also sort of a black box model where we don't really know what's going on. And they're also limited predictable for human health because it's a rat and not a human, and there are slight differences between the species. So we need a solution. And here I'm here to tell you about the solution we thought of to use computational modeling as an option. And let's also discuss sort of the concept of the virtual human. So of course we can't solve everything at once, so we have to start somewhere. So we did a case study on the neural tube closure, and the neural tube is the basis for the spinal cord and the central nervous system. It's an event that happens early during pregnancy, at the end of the 3rd week. A lot of different chemicals can act on it because it's a complex process. A lot of different things happen. So many chemicals can interact with the biology of this process. And it occurs very often when neural tube closure fails. We speak of a neural tube closure defect. And for example, spina bifida happens every 2.5 out of 10,000 cases in the US, which is one of the specific variants of neural tube closure defect. For example, here you see an image of spina bifida, where it's the lower back is open and you see sort of the spinal cord protruding out of the body, which brings very severe handicaps with it. People are paralyzed from the leg down, have bladder problems, have a short lifespan. So of course, we want to make sure that these things do not happen. And there's no direct test available for risk assessment besides for more animal studies. So we want to develop a new test strategy for this case study, and that will be like a computational. But to model something, we need actually to understand it first. Otherwise, you can't really model it. So first, we want to really discuss the start. Okay, what do you need to do when you build a complex agent-based model? If anyone has any questions, by the way, you can just, I guess, raise your hand and then ask, or ask in the chat. So then everyone will be on the same page during the presentation. So first we'll introduce, like, when you build a complex computational model, where do you start and what can you use for this? And then we'll dive into what I developed and what I built and show the results I achieved with the computational model. So everybody starts with a blueprint, whether you build a house or interior or some like spaceship kind of thing. We need sort of blueprints for everything, including biological processes. Here you see an image of the neural tube closure. And we don't do it blindly. There are some frameworks that can help us with these kind of things, where we have, for example, the physiological maps or adverse outcome pathways. And I assume people are not familiar with it because it's more like in the field of toxicology. So I'll shortly introduce them, the concept, and also how you could build one yourself. So what we mean with physiological maps is basically the idea of charting physiology, sort of the biology. We can use different tools for it. For example, you can do it in Cell Designer, and it uses the System Biology Markup Language, SBML. So we start with the first version of this physiological map of neural tube closure, which was manually created. But as time went on, you get sort of like novel tools to create literature for your literature searches, review projects. For example, SysRev or AsReview, if people are not familiar with this. And the concept here with these like literature searches or these kind of tools is that you do a web scraping or a literature search on, for example, the PubMed database. You put a big stack of all the information you find, and you sort of make questions with your abstracts. So it could be labels. Is this relevant for me? Yes or no? Or does it contain a certain species or a process of interest? You then sort of annotate those things. You distribute this big stack of paper between your peers, or if you're like a PhD, you have to do everything on your own. And along your shoulder, there's like a machine learning model, an AI model, looking at the data and what you do as well. And the AI model takes like part of the review work from you. So it's quite a big time save. And what you then end up with is sort of a prediction where the AI model really tells you, all right, this is the unreviewed part. This is very relevant for your project as well. So you can take this along with your model building, with your physiological map creation. Here on the right side, you see an example of like labels. For example, do we want to include it? It's the review, species, experimental design. What level are we talking about? Is it about genetics? And these are all very useful tools. It's a very useful tool to really get data quickly, get information quickly, to then be able to put into your computational model. So really the first start of what I want to build a computational model, where do I start with? And I would recommend start with like the knowledge, like what I'm actually going to model. Otherwise you'll run into a bunch of different difficulties further down the line. So we started to chart physiology using this concept with like one linking pin sort of in the middle, which is resinoic acid, because we know that resinoic acid is very relevant compound for the neural tube closure. Just move myself a little bit away here. All right. And what you then get is sort of lists of all the processes involved in our process of interest, neural tube closure, genes, and also identification of the most critical and important molecular and cellular functions, which are really the definition of critical steps. And if we talk about lists, we really mean lists. So this is basically what you could get as an output, all the different processes are relevant, that are useful, and that you can then integrate in a computational model. And these physiological maps are sort of anchored in Go. People are not familiar with Go. It's sort of a system where we standardize different gene and protein names using specific ideas, which also makes changing our results or information very convenient, because you know that you talk about the same thing, because you use like code terms to communicate. And then we can host these kind of physiological maps in Minerva, which is an online tool. You could view it as sort of Google Docs, but then you can't really work in it side by side together. But people can comment and chat within this tool, so it makes really very easy to correct certain sort of, for example, things that you not agree with each other with. So you can discuss it, and it's very convenient for that. Well, this is basically how like the physiological map would look like. This is an example of the liver, which is already nicely created with a nice cell on it. So the idea with this physiological map is that it's like modular also in structure, and that you can easily navigate these kind of physiological maps. And you have different layers. So you have like the main process, then you can, and things are interactable, clickable, and then you can click on one of the processes, which are then also divided again in different processes. So it's extremely deep and it can be very detailed. So for agent-based models, if you want to model something similar, of course, this level of complexity is not necessarily needed. But if you want to envision these kind of models and where you can go with it, for example, to sort of like a virtual human concept where you try to model all the biology in one place, then you really need this high level of detail in your model to then build models on top of. So this is basically how it would look like. You have like your big physiological map model, and then these events are clickable, which then result to different specific processes. So that was basically the concept of physiological map, one of the anchors in building these computational models. If there are any questions about this, let me know and I can answer them. And then another part is adverse outcome pathways. This will be much shorter because basically how you can achieve an adverse outcome pathway is a bit similar to physiological map. You start with like literature search. You use different tools for it, like SysRev or AsReview, and then accumulate the results in a similar manner, but you look at different things. And basically what you look at is sort of a stressor and key steps in such a pathway. So the idea here, from a toxicological point of view, is you have a molecularization event, a first block, a chemical stressor that then interacts with your biology, and it falls down like dominoes. So that hits like certain large molecular interactions, which results in responses not functioning well, that cause damage to organ responses, which then yields an individual response, for example, a neural tube defect. And we're also working on this in OnTox. It's poorly visible as I can see, but the concept here is that the general idea here is that you really look towards what would happen if things go sideways. And you can also then, based on your literature search, see, okay, which events are most critical. For example, everything that happens here in this example of liver metabolism feeds through sort of this intracellular lipid accumulation part. So if you want to build a model of the liver, then it would be weird to not really have the intracellular lipid accumulation as a centerpiece. And now you really know, okay, what are the essentials we need to model, and which processes are less essential, and really helps you to narrow down your computational model if you want to, and also helps you to see, okay, how extensive do we need to go with it, and what is the minimum product I need to achieve to be able to model diseases, for example. So to recapitulate it a bit, so based on when we build computational models, we're just guided by sort of biological knowledge with different frameworks, and really try to structure biological knowledge because it's vast and it's complex. So we need to have some help with it to be able to find a way through it. And we have our physiological maps, which defines healthy neural tube closure. And with physiological map, we basically map the molecular events that lead to important cellular events. And here you have an example of how physiological map would look like then. And within these physiological maps, these cellular events are this specific process. For example, you have this process of dose-Dutch hinge point bending, and all the events that lead up to it, all the molecular events that lead up to this cellular event. And on the other hand, we have the AOPs, where we link a disruption, a molecular disruption, to a certain phenotypes. And it really highlights which cellular events are most important. For example, if we inhibit BMP4, a critical player in the physiological map that's shown here, it leads to various other events disrupting this cellular event or those types of hinge point bending, which then can lead to a neural tube defect, which we want to make sure that doesn't happen. So when we connect molecules to outcomes, we basically see, all right, we have a successful hinge point formation described by a physiological map, and leads to an outcome, the neural tube closure properly formed. But then in the flip side, we have a disrupted hinge point formation, our molecules, BMP4 and these cellular processes. Again, we connected to outcomes. And in this case, we see that our hinge point formation is disrupted, which is these sort of bending points here. So they're not here, as you can see. And here's also where age-based modeling comes in. Because we really thought about like top-level information. We talked basically about graphs and boxes and arrows, but you don't really visualize it. When you look at phenotypes, you don't see a box connected to an arrow. You see really like these kind of shapes in tissue that happens in biology. And with age-based modeling, we can visualize these phenotypes because cells follow basically rules derived from gene expression. The gene expression we capture in our frameworks, and this collective cell behavior leads to these tissue-level outcomes, as you can see here on the right side. And that's why we simulate these models. That's why we build these models. And probably you guys are all familiar with it, with CompuCell3D. So in CompuCell3D, we represent the age-based model as individual agents. Here's an example. Maybe Julio has showed it already during this workshop. And this is how I represent it. This is how it looks in CompuCell3D. And when we run it, it sort of looks like this. But to this cell, there's not really behavior attached. What we want to do is really simulate cell behavior during neural tube closure. So really the computational modeling of this process. And as you can see with the neural tube closure, if you look at this process, a lot of different things happen actually. Like at the first part, you start with like a sort of a flat layer, flat ectodermal layer with different protein gradients going on. And cells start to proliferate. The flat layer starts to sort of go up. It starts to form and bend and shape. You get different physical forces playing a role in as well. The pushing of these cells, the sort of scooting of these cells. And at some point it fuses and different cells migrate away. So when you look at this vast process where like a bunch of different things are happening, you need to ask yourself the question like, what is essential? These are these key events, which we derive from the AOPs, such as the key events, cells attaching to this part, mean hinge point formation, bending of these cells, those with hinge point formation, those cells migrating away, and how are deregulated. And for that, we look at the physiological map. So we deduce a number of key events during neural tube closure. We're not able to simulate everything all at once because it's too big, too complex. So we really focus on a set number of key events that are most critical. Which are floor plate formation, dorsolateral hinge point formation, and the neural crest cell delamination. And we extract the gene regulation network from the physiological map. So we built like this huge network, right? And it has way too many interactions to properly simulate, or of course you could try it with Boolean modeling, but we start a little bit of a simpler approach. And we use this as a template for a gene regulation network in our agent-based model. And of course, we are interested in key events. We discovered that are very critical, which are mean hinge point formation over here, dorsolateral hinge point bending, and neural crest cell delamination. And basically, from these key events, we try to move backwards. So first, look at the direct regulators of these key events. Look at the receptors and regulators that regulate those direct regulators. Let's look at the protein gradients that interact with other receptors on our cells and the processes that make these gradients and secrete these proteins that interact with our receptors. And based on these critical components that regulate our key events, we derived sort of this gene regulation network. And this gene regulation network is basically the engine that powers the model, a large part of the model. And it consists of different things. You have like genes, proteins, receptors, key events, compounds, such as ATRA. And it consists of like activation mechanisms and inhibition mechanisms. So the way I coded this in CompuCell3D is actually just in a steppable. And what I used are hill functions, very simple hill functions scaled between 0 and 1. Because we don't really know the exact details of these processes, the exact mechanisms of these secretions, the exact amount of these secretions. We just know that something activates or inhibits. So by really taking a step back and make it very as simple as possible, we are able to mimic a lot of the stuff required to get this model to work, which I'll show later. So after integrating this gene regulation network within our different cells, we can then model the neural tube closure. As you can see here, compared to the images, it starts with like a flat layer. Cells start to divide and move up. Cell starts to migrate to the middle. You get like those sort of hinge point formation. Cells fuse and different cells take off and move away. Now, if we visualize this in, because we have our gene expression coded in CompuCell3D, so we can also visualize this. We see here all different genes that are encoded in our model really show distinct expression patterns. See a lot of stuff going on. You see BMP4 coming up here on the sides, together with PAX. You see GLI2 expressed down below, sonic hatching brain going on. So a lot of different things is happening. Yet we used a very simple approach to model these kind of gene interactions, actually. Then if we compare to actual in vivo data, in situ hybridization data, we see that the patterning of a model is very well reflected compared to actual biological results. See here, for example, sonic hatching clearly expressed here in the bottom. We see pitch sort of moving to the halfway down. We see PAX3 really on the top, expressed in specific cells, being before secreted from the top level here. And we managed, even with such a simple mathematical approach, to really recapitulate biology as necessary to be able to model this complex process. So this gene regulation network drives a large part of this complex cell behavior, different protein secretion, formation of the floor plate, somites, physical forces, local cell domination, and apical constriction. So with such a network, we're really able to steer the biology in a way that it goes in our biological observations. So now I want to, these are all the processes that are involved in the, that our gene regulation network steers. So now I want to dive a little bit deeper into one of the processes to tell you guys about how I exactly modeled it. So you have like a general idea what's going on and how I approach this kind of processes. So going to apical constriction, and apical constriction really is the formation of sort of bottle-shaped cells. Here you see a more complicated simulation of apical constriction I built in CompuCell3D. And it's an essential mechanism, and it really happens here on the dorsolateral hinge points, here on the sides, and the middle at the median hinge points. As you can see, as apical constriction processes, you really are able to make like different shapes and forms of a flat layer of cells, make them a circle, for example, with just the apical constriction mechanism. And the way I code apical constriction in the model is actually quite simple. Julio helped me with this as well. And what we basically did is we reduced the apical size of the cell volume. So this is the apical side here. Apical is that pinkish, lateral blue, and basal green. And first, because cells become like bottle-shaped, you start by reducing the apical cell volume and increase the basal cell volume. So in similar amounts, because the cell doesn't get bigger or smaller, it's just that volume changes over time. To do this, we need to split one cell into different compartments, an apical, lateral, and basal side. And we need, of course, ensure that cells do not break apart. So we use like the focal point plasticity links. You guys probably had it in your course already, to keep cells intact, and then to increase the distance between the cells, because cells not only become bottle shaped, they also become elongated if you compare these cells here to the cells over here. And we elongate cells by increasing the distance, the target distance between the apical and the basal cell, and it pulls the lateral cell with it, really getting this bottle-shaped cell that we see when we watch apical constriction under the microscope, for example. So then, of course, what's important is the mechanism of apical constriction. We can code the behavior, but it's triggered by a specific mechanism that we, of course, got from our literature searches. So apical constriction requires intermediate levels of BMP protein, and it's inhibited by sonic hedgehog. So with sort of these simple conceptual rules, we are able to then model apical constriction, where we assume, because we know it requires intermediate levels of BMP, and it's inhibited by sonic hedgehog. So you can sort of calculate the chance of apical constriction to occur for a cell, which you would, for example, and these are not actual graphs, it's just like a representation, model a probability of apical constriction on sort of a shape that's like this. Like it needs intermediate levels, so then the probability is highest at intermediate levels. Times, because sonic hedgehog inhibits it, sonic hedgehog concentration, where sonic hedgehog is low, or where sonic hedgehog is low, the probability becomes highest, but when sonic hedgehog concentration increases, the probability becomes near zero. So it's really a probabilistic concept, what we introduced here in this computational model. And if you then run the model, you would see apical constriction gradually increasing, where the red ones are currently apical constricting and the blue ones are not. You see that over time, based on these gradients, apical constriction changes location and becomes more over time. What you basically see is cells start to flicker. And when a cell goes into apical constriction, it goes one step towards maximum apical constriction. And when it's blue, it goes one step down towards minimal apical constriction, which is just a normal state. And this concept is basically modeled using a set of different equations. So I approximated the intermediate largest apical constriction chance using this formula. Then you model it by sonic hedgehog, which is basically a Hill function for inhibition. You calculate the rate, so combined probability of those two, and you correct with the Poisson constant for time. So another thing that's very important in some of these like agent-based models is cell contact. You guys probably played with like this model. When I think 3 years ago started my PhD, I also did the CompuCell3D model, and this is the first one we run, I believe. So with cell contact, we can really simulate cell-cell adhesion, and it's a proxy for cadherin-mediated adhesion, which basically means that a lower value cells will clump more. And you can basically make the spatial organization using only this concept, because you have all these different cell types. These are like seven or 10 cell types. And you really want to mimic our human biology. So then we have our computational model here, we have our like different states, and we put them next to actual human embryo sections. What you see here is that there's a lot of similarity. So you hear this neural plate structure. You see here different cells in sort of a different mesenchymal cells and sort of like fluid. They start to clump together in time, forming structures called the Somite. Well, the rest continues here. You see like the DOS range point forming, the bot-shaped cells here, which are a little bit blipped out here, and then the fusion on the top. And really, just by playing with this like cell contact parameter, like this concept, we're able to shape and form our model to reflect biology. And then you can also compare, for example, to a mouse, which looks a little bit different, but still is highly similar. So, like this concept, where we have like cells in the fluid and cells clump together, is actually quite simple to achieve. Where you create the extracellular matrix, which is in yellow, between the mesodermal cells at the start, and you just make the contact energy between the ECM and the mesoderm very low, 0. So that's why it gets this fragmented portrayal. And then you can differentiate the cell to a different mesodermal cell to a different cell type, for example, the Somite cell, which then increases the contact energy over time, making like these fusions over here, right? You see like these more stiff and rigid structures, which again, if you compare it to biology, is highly similar if you compare these mesenchymal cells over here with these more rigid cell shapes. Of course, our cells in our simulation are not that elongated, so we have a much more simple, more simplified structure, yet it really achieves the concept we're trying to achieve, a stiff and rigid structure that's able to push up these neural folds. So if we then look at this and place everything together, we get sort of like a dynamic systems model, because we have our gene expression, which then results in a cellular response, which then results into sort of tissue morphogenesis, which then results in developmental outcome. So then you can ask, like, all right, we built this model, but what can we do with it? So what we can do is, of course, our goal is to model neural closure defects. And we have this gene regulation network, and we can basically adjust the fold change of the key genes in this network to simulate potential perturbations. Let's say chemical inflicting an inhibition effect, which then leads to a neural tube closure effect, as portrayed here with our AOP. And then we need to validate our computational model against known biological data. So here we have our normal simulation, sort of our wild type, where it's healthy. And when we run this, we get like our healthy neural tube, where it's fused properly. We then remove, for example, BMP4, the chemical you just saw. And what we then end up with is that compared to the wild type, the dose-load hinge points don't form so well. And actually, you end up with a completely different structure. Very similar as the images we earlier saw, because BMP4 really is essential for the formation of these hinge points. You can also look into other different proteins, let's say sonic hedgehog. That also has a premium function during neural tube closure. And although you can see that those hinge points still form, you see that there are other issue occurs where the floor plate, instead of being that stiff and rigid structure, really becomes sort of like a cell ball that doesn't really help that well, given the proper spatial organization the neural tube needs. And again, if you compare this to in vivo data, you see a very similar pattern occurring between our computational model and what's observed in biology. Another option could, for example, be a noggin, which is an inhibitor of BMP4 and has an important role during neural tube closure as well, as that you see that the midline sort of fuse together. Again, very similar as what we find in biology. And here you have the images not moving, so they're stiff, so it's easy to compare. We have a wild type, knockout, and or knockout situations, you can see that each of these simulations really look like their biological counterparts. But not everything is the same, not every simulation. And because, of course, the CompuCell3D age-based models are stochastic, so they give multiple different outcomes. And it's actually a big benefit for it because risk assessment in toxicology doesn't have one single outcome. We always speak of, oh, this chemical increases the chance for a certain defect. And that's one of the strengths of our computational model in CompuCell3D, because we can really generate many different possible outcomes and also plot them as a sort of a quantification prediction, as a probability, a chance of defect. And if we run our computational model a bunch of times in our different knockout scenarios, we see, for example, even the control has like a small chance of getting a defect. And all the other situations have like a probability of having a certain outcome. Let's say a midline fusion defect, which is here, or a closed neurotrope defect, or an open defect. And we can also then, instead of not removing a gene, overexpress a gene, which again then yields a different pattern, a different types of neurotrope defects. So where do we go from here? I hope you guys agree with me that computational modeling shows promise. And now what I want to do is really use this computational model to go towards a chemical validation application, and rather simulating knockouts or hyper or overexpression, really simulate chemical data in our age-based model. And how we want to integrate this is basically use simulation outcomes to predict a hazard. We have, for example, our ADME model that feeds into a set of different tests, in vitro tests, that then generate parameters that we can put into our virtual human in silico model to really generate a safety profile. So it's maybe one small step for humans and animals, but a giant leap for animal-free predictive risk assessment. And with these kind of simulations, we can help protect human health through in silico models. And for animals, we may be able to give them other tasks besides being experimental animals. Maybe they can program computational models for us. I didn't do it alone. I got help from my promotion team, Dr. Harm Heusingveld, Albert Piersma, and Juliet Lechler, as well as very important external advisors, James Glacier, Thomas Knudsen, Richard Spencer, and Giulio Belmonte. And I thank you for your attention. If you want to know more, you can contact me. This is QR is the link to my latest publication. And if you have any other questions, you can shoot me an email. Are there any questions right now?
Thank you, Joe. Suvani, did you want to ask your question? You can unmute and ask.
Hi, am I audible?
Yes.
Yes. Yeah, so thanks for the presentation. It was really insightful. I actually wanted to ask that have you used Boolean logics or ODs to put the signaling networks into cell? Because I think you mentioned that interaction details were not like explicitly there in the network profiles.
Great question, thank you. So in this current state, I use Hill functions to approximate the gene regulation network, but together with some collaborators, we're currently translating the physiological map towards a Boolean model and want to try to integrate it with the agent-based model. So we're currently working on it, and it looks very promising.
Thank you.
Mark, would you like to unmute and ask your question?
Well, my question was kind of solved during the presentation, but initially you showed many chemical fields that you were using in the simulations, and then you showed the simulations, and I was asking how did you couple both things, the different cell behavior that we observe and the chemical fields? but I think it has been quite well explained with the example of the apical constriction.
Right. I can just clarify maybe a little bit more. We have the chemical fields, and then the chemical fields touch a sort of a receptor on the cell, and the receptor translates the signal to the rest of the gene network, also via Hill functions. So to maybe clarify a bit more.
Thank you.
Okay, does anyone else have any questions? Please just unmute and feel free to ask.
Hi, I have a question, please. Thank you for the great presentation. It was very insightful. I wanted to ask, in the context of toxicology and risk assessment, how this model that you built could be integrated in the context of risk assessment. I mean, my background is toxicology, and I'm taking the course just to get into that. But I'm not sure right now. Of course, or at least myself, I am a beginner, so I would like to better understand how this could be used, if possible, to make some predictions for chemicals.
So thanks for the question. There are different ways to look at it, so to say. Like there's not one golden rule or road. Like there are multiple roads to Rome concepts, you know. So how I envision it personally is you can sort of make like how do you call it? Choose diagram, is that the correct word? Where you say, all right, we have this chemical with a structure. So we can, for example, first do prediction or QSR-based analysis. That results in potential binders of the certain chemical. Do we have then these binders, these proteins in our computational model? And then we can then run the percentage of how sure the QSR model is that it is a binder as a parameter in the model. Say it is 80% sure it's a binder. We can run an 80% reduction of certain gene expression of genes in our gene regulation network. Another way is to really go into the in vitro part, study the chemical in vitro, extract parameters from it. Let's say you see a high increased level of cell death, so you add a higher probability of cell death to your computational model. Like it's not really, this modeling frame is not like the holy grail, it will solve everything, but I think it can really be used as a centerpiece, and every study you will do anyway to study the effect of a chemical. You can also integrate this information in your computational model to then improve it over time. Does it answer your question?
Yes, perfectly. That was the piece I was missing until now to try to incorporate this type of tool to my work. Thank you.
All right, great.
And great presentation and great work, of course.
Okay, well, happy to hear it.
Okay, does anyone else have any questions?
I have another question. Maybe in this kind of informal presentation I can make. Because I'm starting to use Southput model, and I have also many parameters. And I was wondering how you parameterize this huge model where I guess there are tens or hundreds of parameters. Did you make parameter inference from many experiments, more parameter exploration?
Yeah, so basically how I achieved it is the most important thing in these kind of large complex models is really like, okay, well, I'll ask the question like, what am I exactly going to model? So we look at these kind of different frameworks, like, okay, you don't really have to build a physiological map in the detail that we did, but you can like explore, okay, what are the events that happen during my biological process, and which events is most important. Because if you know like which event is most important, then you can really focus on those to parameterize those processes well, so that that works as best as possible, and then add other parameters to like make it work, so to say. So you have like your key events that really need to be correct, and side events that are nice to have but don't have to be as precise. And then you can do parameter sweeps. If you, I think I have like the script I used is with publication. So where you can just run different instances of the same model and then see how it behaves. does it behave like biology? As I put the images of, for example, my screen still shared, right? Then just go to that slide. Example, let's say this one. Does my model look like biology? A very simple question. You can just find the image of your process, or if you do cell culture yourself, just put it on the microscope. Does it look correct? Very pragmatic, maybe. Another thing is, all right, look at gene expression. And in these cases, it's way more back than I expected. Yeah. So in these cases, like I don't know how high all these values are, right? I just know that, for example, BMP4 is high at the top. So it's most high at its point of origin and least high on its farthest away point of origin. So then with that sort of pragmatic assumption, you can then code it to sort of reflect this behavior. Because in these healthy situations, it should look like this. It shouldn't look like in any different way. So with these sort of simple rules, comparison biology, you really already are very close to approximate correct parameterization. And then it's just a question of trial and error, run many different parameter sweeps, and then pick the ones that just show the best results, highest probability in health situations, and then also validate it with sort of the knockout studies I did and see does it reflect the behavior as it should in biology. It's basically how I approached it. There are probably people with different approaches because you can do it in many different ways. But I hope this helped you a little bit with your question.
Yeah, very clear. Thank you. Great talk.
Great.
Any other questions?
Yeah, I was going to say, any other questions? Otherwise, let's thank Joe for his wonderful talk, and we look forward to your future work. And that will wrap up today's presentation then, and hope everyone is enjoying the CompuCell workshop so far. Thank you, everyone. Thank you, Job.
Thank you. Bye.
Bye.